Hey guys, it's Jaelin behind the camera.
And Chanel demonstrating the process of meiosis.
And today we will show you guys how meiosis is structured and the process it undergoes to produce genetic diversity in organisms. Meiosis occurs only in eukaryotes. In animals, it occurs during the production of gametes. In plants, it takes place when spores are produced. Meiosis does not occur in prokaryotes. They produce by binary fission. Okay guys, so the first stage of meiosis is interphase 1, where the DNA in the diploid nucleus replicates itself. The identical copies of the chromosomes stay with one another at first dubbed sister chromatids. The centromeres with sister chromatids intersect. The next stage is prophase 1, where the spindle fibers form. And we're over 90% of the time cells spend in meiosis. During prophase 1, the two members of each homolog pair become intimately associated along their entire limbs. They synapse, meaning the joining of homologous chromosomes, which produces tetrads, also known as bivalents. This is to show that the two chromosomes of each homolog pair adhere to each other during this phase of meiosis. But the appearance of a tetrad changes in the different substages of prophase 1. Early on in this phase, the two chromosomes of which a tetrad is composed fuse together so tightly that they cannot be distinguished from each other. Only later do they become separately visible. In the upper diagram, two tetrads are represented as two X-shaped chromosomes associated side by side. After prophase 1, crossing over is complete. The tetrads move to a plane called the metaphase plate, halfway between the two poles of the cell. Next, the spindle fibers attach to the centromeres of each chromosome. Both kinetic cords of each sister chromatid pair are turned toward the same pole. As a result, both kinetic cords attach to spindle fibers to the same pole. So, towards the end of prophase 1, the nucleus precipitates so the chromosomes can get out to the cell. Crossing over can occur between homologous chromosomes, which is likely to occur at several different points, resulting in chromosomes that are mixtures of the original two chromosomes. The next stage is metaphase 1, where bivalents become aligned in the center of the cell and are attached to spindle fibers. The chromosome alignment in our display is from a cell with the diploid chromosome number of 4. Thus, this stage ends with the homologous pairs. Then we have the stage anaphase 1, where the cell starts to lengthen. The two homologs of each chromosome pair separate and move toward opposite poles, drawn by the microtubulates of the spindle apparatus. This contrasts with mitosis, where the sister chromatids of each homolog separate and move toward opposite poles. Here, the chromatids remain together as one complete replicated chromosome. As you can see in our display, this is the result of anaphase 1. A key difference between mitosis and meiosis is the sister chromatids remain joined after metaphase and meiosis 1, whereas in mitosis, they separate. You will see a difference in anaphase 2, where the sister chromatids separate, whereas in anaphase 1, sister chromatids remain attached as shown. The next phase is telophase 1. At each pole now, there is a complete haploid set of chromosomes, but each chromosome still has two sister chromatids. A cleavage furrow appears, and by the end of this stage, the parent cell has divided into two daughter cells. This separation of the cytoplasm is called cytokinesis. In some organisms, nuclear envelopes appear briefly at this point. The intermediate stage is called interkinesis. But in others, the daughter cells begin immediately to prepare for the second meiotic division. As a result, two nucleuses form because of the division of the cytoplasm. In many organisms, the two daughter cells produced by meiosis 1 begin immediately to prepare for the second meiotic division. In others, however, nuclear envelopes temporarily appear and enclose each of the two separated sets of chromosomes between telophase 1 and prophase 2, and there is a period of rest. This period, during which envelopes are again visible, is called interkinesis. Each chromosome is still composed of two sister chromatids. The period between meiosis 1 and meiosis 2 is sometimes called interphase 2. Since there are two divisions of the meiotic process, we will now move along to meiosis 2. So prophase 2 begins with the two daughter cells produced by the first meiotic division. As in prophase 1, the chromosomes are condensed. Because there was an interkinesis, the nuclear envelope dissolved. The centrosomes have replicated and are moving toward the poles. Again, the chromosomes are not yet attached to the spindle apparatus, which is growing outward from the centrosomes. Moving along to metaphase 2, where the two daughter cells produced by the first meiotic division, which are known as secondary germ cells. The spindle again draws the chromosomes to the metaphase plate. This time, unlike metaphase 1, the two kinetochores of each centromere bind to spindle fibers from opposite poles, as in the mitotic metaphase. This results in the separation of the sister chromatids of each chromosome during the next phase of meiosis, anaphase 2. During anaphase 2, the sister chromatids of each chromosome separate and move toward opposite poles. Once they are no longer connected, the former chromatids are called unreplicated chromosomes. As the chromosomes are dragged along the spindle apparatus, their arms can be seen dragging along behind so that the chromosomes form a somewhat V shape. The poles themselves move further apart as cytokinesis begins and the cell lengthens. Because crossing over occurred during prophase 1, the sister chromatids will have a genetic variation. Lastly, we will have telophase 2, where the chromosomes reach opposite poles. Cytokinesis occurs, the two cells produced by meiosis 1 will divide to form 4 haploid daughter cells, and nuclear envelopes will form, which you can see are lightly drawn on our display. When the two cells are entirely separated, their nuclear membranes are fully formed and meiosis will be complete.
Thank you for watching our version of the meiotic process.
Peace.
Chanel and Jaelin.